Hello, everybody. I'm Professor Alice Roberts, Professor of Public Engagement at the University of Birmingham, and this webinar is part of our Bringing Birmingham to You series, organised by our wonderful alumni office. And I'm delighted to be inviting around 700 alumni and friends today from all around the world. This is the second Mapping the Virus seminar, following the first, which was back in November. Now we're a year on from the first national lockdown, and new variants of COVID-19 have emerged, vaccines are being rolled out, and countries around the world are in various stages of easing restrictions or bringing them back in. Here in the UK, we now have schools fully open with a roadmap for opening the rest of society. Throughout the pandemic, experts from the University of Birmingham have been at the forefront of testing, genomic sequencing, and studies into vaccines. Before I introduce the panel, this is an interactive webinar. We're not just going to talk at you. Some of you I know have submitted your questions in advance, but you can also submit questions live throughout this webinar using the Q&A box that you should be able to see on your screens. If you want to ask your own question in person rather than having me read it out, let us know that in the Q&A box. And if you do come on to ask a question, then we will turn on your camera and your microphone for you at the appropriate time. If you don't want to ask your own question and you prefer that I read it out, that's absolutely fine. And you can even ask your questions anonymously as well. Please feel free to use the chat function through the webinar as well. Okay, then it's time to introduce our illustrious panel. First, we have Professor Alex Richter. Alex Richter is an alumna of Birmingham Medical School, and she's now Professor and Honorary Consultant in Clinical Immunology. She's been leading a team of experts from the university to develop a SARS-CoV-2 antibody test focused on improving detection in individuals who have mild disease. And she's also worked as a national scientific advisor on virus testing. Alex, what are you doing right now? Alex, you're muted.
Classic mistake, isn't it? So thanks, Alice. So the focus of all my research at the moment has switched from looking at immunity to natural infection to immunity following vaccine studies. So we've got a number of collaborations within the Birmingham area, but also nationally, trying to look at different groups of individuals and patients and how they respond to the vaccines, the different doses and the different types of vaccines. So an exciting time.
Thank you very much, Alex. Next, let me introduce Professor Alan McNally. Alan is Professor in Microbial Genomics, and he is the Director of the Institute of Microbiology and Infection at the university. He looks at the evolution of viruses by studying their genomes, and he's also interested in drug-resistant bacteria. In April 2020, Alan became the infectious disease lead at the government's first flagship COVID-19 Lighthouse Lab testing facility. And he's currently heading up the COVID-19 testing lab at the university. It sounds as though you're still fairly busy then, Alan.
Yes, we still have some involvement in Pillar 2 testing. So that's testing of people with symptoms in the community. And recently, actually, a lot of my attention has turned to using lateral flow tests for our student and staff on campus to try and maximise how safe campus is for students and staff using the campus. So a lot of lateral flow testing recently as well.
Thank you very much. Well, last but not least, we have Professor Nick Loman. Nick is Professor of Microbial Genomics and Bioinformatics in the Institute for Microbiology and Infection. He uses cutting-edge genomics and metagenomics for diagnosis, treatment, and surveillance of infectious disease. And he recently helped establish real-time genomic surveillance of Ebola in Guinea. But what are you doing at the moment, Nick?
Thanks. We've been working on sequencing SARS-CoV-2 since the very start of this pandemic in Birmingham. And so far, Birmingham's contributed about 5,000 genomes to the Coronavirus Genomics UK sequencing effort. In the last, since kind of Christmas time, the world's kind of gone genomics crazy and got interested in genomic epidemiology, understanding how the virus is spreading around by looking at its genome, and also got very interested in the emerging variants. I've been advising Public Health England and how we can best discover new variants and trace the variants that are circulating globally. So we've been spending an awful lot of time peering into genome sequences and trying to spot the next big variant threat, and thinking a lot about how the virus will evolve in response to the vaccination program that's going so well in the UK.
Thank you very much. So there are the panel, and we have some questions that have been sent through from you already. I'm hoping that some of you may be there to ask your questions in person, and I wonder if I can invite Simon Rhodes to ask his question. Simon, are you here with us? I think he is here. Hopefully he will pop up in a second and ask his question.
I am here indeed, yes. Hello.
What's your question?
Can you hear me okay?
Yes, I can.
Hi,
Yeah.
thank you very much for having me. I'm an old colleague of Alex's, so it's very exciting to hear her speaking. Thank you. Hi, Alex. My question was, and it's probably more of a theoretical one, but with the obvious need for vaccines not quite met by the demand, and there's a decision to delay the boosters, which isn't the question. There is talk that for people who've had proven COVID, and particularly if they're antibody positive, might only need one vaccine. That the original infection might class as their first vaccine in a way, and then the single vaccine they had almost is like a booster. Is there any merits to looking at that as a way of just sort of spreading the vaccine supply around? That was the question, really.
Hi, Si. Yeah, it's a great question, and I call this the French way, because this is the strategy they went for. But actually, from our early vaccine data, there seems quite a lot of merit to this argument and this strategy. So the early vaccine results that we're getting out now, we've split up and looked at the quantity of antibody produced post natural immunity and post infection. And post vaccination, if you haven't had an infection, your antibody rate is still higher than with natural infection. But if you've had natural infection and you have your first vaccine, your rates might be 10-fold or 100-fold higher than natural infection. So you're getting incredibly high antibody levels with this first dose, but it, as you say, is a boost. I think from a policy point of view, the UK government's probably too far down the road with most of the at-risk groups to go away from the single dose, double dosed. But I think it's going to be really relevant going into the autumn and deciding what we're going to do with boosting. And those that have had national infections and those that have pre-existing good antibodies are probably not going to need boosting going into the autumn.
Thank you very much, Alex. And do we know how long antibody protection lasts for?
It's the million-dollar question, isn't it? Because we want to have neutralizing antibody which protects us from the virus re-entering our cells. What we've found from a number of cohorts, so we set up a healthcare worker cohort here at UHB and also a dental cohort. And so data from about 4,000 members of staff has shown us that at 6 months, with antibodies from natural infection, you keep hold of those antibodies for a good 6 months. So 2/3 of people keep their antibodies. But of those that have lost their antibodies, it doesn't mean that they're not immune, because remember, the cells that make the antibodies are really long-lived. So these memory B cells that make the antibodies are really important for reactivating with reinfection. But what we have found with those two cohorts is that it is the patients that have lost their antibodies or have the lowest level of antibodies that are more likely to get reinfection. So the first antibody tests that we had were what we call qualitative. So they just give a yes or no answer. Whereas the assays that have been coming out since Christmas are starting to give us an actual number of how much antibody you have. And we'll be able to translate that hopefully over the next 6 months or so to know what is a protective antibody level.
So do you envisage then, just, I mean, going back to the question from Simon, do you envisage that there will be antibody testing as part of the vaccination programme as we get through into the autumn?
I think this is something that the government's grappling with at the moment, and this is one of the questions that's been asked. What does our antibody testing capability need to be, as well as our PCR testing capability? And a lot depends on how we're going to use those antibodies. So what we tend to do with research is we try and get a representative sample of people and see how that representative sample reflects the whole population. So if we know that the great majority of people with a certain level after vaccination have great antibodies, then there's no point in testing. So like after the kids have their childhood vaccines, or you have your flu vaccine in the autumn, we don't check your levels to check that you've responded, because we know that the majority through previous studies have responded. So we're hoping to get that information first. But if we find that that response is very variable, then there may well be a role for antibody testing.
Thank you very much, Alex. I wonder if Alan or Nick, you want to comment on this question, or we'll move on to the next one. Okay, we'll move on to the next one. It comes from Heather Allen. Heather, are you there?
Yes, hello.
Hello, what's your question?
My question is, how much of the world would need to be vaccinated for the risk of infection to be over, and how long would that take?
That's a great question. Thank you, Heather, because at the moment we are in a situation where individual countries very much seem to be tackling this as individual countries. Does anybody have an idea? Who would like to start with Heather's question? How much of the world does need to be vaccinated? How long is it going to take? I'm going to pick on you, Alan, I'm afraid.
I think the reason we've all sort of laughed and stalled on putting our hands up for that is we just don't know. There are some, there's been herd immunity talk since the beginning of the pandemic, which is incredibly unhelpful. I don't think we really know what the level would be required in order to confer herd immunity. I guess that's the question, is, how many people need to be vaccinated before we could stop and everyone's protected? I think we just don't know. My guess is it's going to be very high for a very simple reason, and that SARS-CoV-2 doesn't just infect humans. And so it's not going to go away when you vaccinate sufficient humans. It's going to go into animal reservoirs. We know it infects mink. We know it infects various rodents. There's been reports of infection of pets. So I think the number would be incredibly high, because we're not going to get rid of a natural reservoir of infection for SARS-CoV-2 when we vaccinate humans. It's just going to infect other hosts. And so the risk will always be then that it can come back into the human population. So my personal feeling is it would be an incredibly high number. And as you pointed to, Alice, I think we really need to think about the numbers of people who've been vaccinated in other countries across the world. There's no point in Britain being 100% vaccinated and all of our neighbours being 50% vaccinated. That's not going to work. So that's a bit of a doom and gloom answer, sorry, but that's my stab in the dark at it.
Yeah, I agree with Alan. I think actually if we think, I think Chris Whitty made this point the other day at a press conference. The only virus we've ever actually managed to eliminate through vaccination has been smallpox, and that took 100 years or more of concerted effort, and that's with a vaccine that was incredibly effective. And so before you even count the very likely animal reservoirs that will be established, I think simply the idea of eradicating the virus from human populations is virtually impossible. So I think the question is going to be more about how much of the world needs to be vaccinated in order to keep the virus suppressed to a reasonable extent, and mindful of the fact that the more virus that is circulating globally, the more we're all at risk of new variants developing with adaptation to vaccination. So we really need good blanket coverage of vaccination globally. It's no good to have individual countries with extremely high levels of vaccination and other countries with very low. That's actually the situation we're moving into, I think, in the kind of European picture at the moment. We are in the UK well ahead of the game which is obviously great news. But of course, that means people start talking about, well, actually, we have a large number of cases in France, which a small percentage of those are of a variant that we're concerned about with regard to vaccine effectiveness. And so start talking about, well, does France have to go on a red list of countries that we can't, we can't have direct, movement from? So, that's clearly not going to work for the sort of society that we want. So it's going to be, I think, we need to prioritize a vaccine strategy that gets good vaccine coverage globally and particularly focuses on the most vulnerable members of the population who are clearly at much increased risk of the virus. But that's not a very quantitative answer to your original question. But I think it's going to take a long time and it's going to be quite difficult. And it's clearly going to be limited by supply and then eventually money to get that global coverage we want.
Thank you.
I think just following up on the quantity there, Nick, I mean, I think when I think about herd immunity or whatever that concept means, I often think about the MMR vaccine and measles outbreaks, because that's where we as a society have a bit of experience of knowing when vaccine coverage drops, we start getting outbreaks. And if you look at the Public Health England data around measles, we see that we start getting higher rates of measles again when the vaccine rates drop below about 90%. So, even if we need something in the high 80s, the logistical challenge alone of vaccinating nearly 90% of the world population without variants, without, animal vectors, it's an incredible challenge.
It is, but I think we should take a little bit of comfort from the Israel perspective at the moment, because I think that their infection rates are looking extremely promising. They have very low rates of novel variants. And I think they're about 60% into, double dose, 60% territory. So they're doing very well with their vaccine program. The flip side, though, is I believe the borders are more or less closed in Israel at the moment. So they've obviously got a slightly artificial situation in terms of importing new variants. So the interesting question for them will be, if they get, they keep the rates very, very low through vaccination. How can they open their borders and keep society going well?
Thank you very much. Moving on now to a question that's come in from Marion Dixie. And her question was, how does the recent link between COVID infection, long COVID, and reactivation of the Epstein-Barr virus change the approach to prevention and treatment? Who'd like to have a stab at that one? Alex.
Well, we're still at the absolute beginning of learning what long COVID is. I think I'm pleased to say it's been recognized, because I remember in the first few months, patients were saying, I've been floored by this, or, significant respiratory symptoms or aches and pains and tremendous fatigue. And, we just didn't know. We were all worried about the patients on ITU, and I think all the focus was that way. And the patient groups were trying to galvanize basically recognition for this long syndrome. And the reason why this long syndrome was important is because it was affecting patients that had mild disease. So what causes it, we're not sure yet. My guess, we're looking at other chronic viral carriage. So we talk about Ebv reactivation. Ebv is one of those viruses that we live with for life. And, I see this in my immune deficiency patients. It's one of the viruses that can reactivate when the immune system is low. Now, the coronavirus is slightly different in that it is an infecting virus and doesn't tend to cause latency. So the way that I think of long COVID is that you've had the infection, but it's triggered some long-lasting, not just immune impact, but impact on all different parts of the body. I'm sure a number of you have heard about ME and chronic fatigue syndrome or post-viral fatigue syndrome. And for years and years, these patients have suffered from this kind of long viral syndrome, which, you know, can be really debilitating. And I think long COVID is going to raise awareness of these viral syndromes and try and work out how we can address them. We've got a couple of grants that we're involved in at the University of Birmingham looking at long COVID. And my little part in the long COVID work is looking at whether COVID infection and other viral infections actually produce autoantibodies, and they affect how the immune system sees itself, and whether that might have a role in long COVID. What vaccine means for long COVID, we're again not quite sure yet. And I've heard from patients, both sides of the story. I've heard some long COVID patients got better when they've had the vaccine. So it almost seems like the immune system has reset itself. But then equally, I know of a few people that have actually then had prolonged symptoms after their vaccine. So I think jury's still out and don't have an answer. Early days, I'm sorry.
But that was a really comprehensive answer, Alex. Although we don't know the solution, we don't know all the answers. We know how to get at the answers, and it is being looked at. Right, I'm going to move on now to a question from Ian Sim, which is a question about virus variants. Ian, are you here with us and happy to ask your question?
Yes, indeed I am. Thank you very much indeed. My question relates to the current design of vaccines, which focus very much on the virus spike protein. And given the emergence of variants, some of which are escaping, some of the data suggests they're escaping the immune response to the existing vaccine, should we be re-looking at the vaccine strategy? Is there a case for thinking that a whole virus vaccine, whether it's killed or even an attenuated virus vaccine, might be a better strategy, or a multi-antigen vaccine might be a better strategy than just focusing exclusively on spike, which I understand gave us, you know, very fast development of a vaccine, but maybe it's its Achilles heel.
Thank you very much for that question. Who would like to tackle this one then? So this is a question about whether a vaccine which targets just a very small part of the virus, the spike protein, could be better or worse than a vaccine which basically involves the entire coronavirus, presumably disabled.
I can have a attempt. I'm not a vaccinologist or an immunologist, but I think it's a really good question, because you could argue that, you know, there are other antigenic components of the virus other than spike, and actually targeting as many of those as possible might give you a more effective vaccine. I think, I believe there are programs along those lines established, but I think the reason that the spike protein vaccines came out so strong is that there's quite a lot of previous work developing vaccination for other coronaviruses, and thinking about MERS and SARS-1, and it's fairly well established how you can make a good spike protein, and also how you can slightly modify the spike to give you a really strong immune response. So it was an obvious one to start with. Something that I think maybe we didn't even appreciate at the time is, of course, the very first, you know, all we needed to start on a vaccine program was that very first genome sequence, and that was released, well, it was done kind of late Christmas time, and it was released around the 10th of January, a bit of controversy that it took that, 10 days to release. But once it had been released, that sets the stage to start developing, you know what you're dealing with, and you know the sequence of the spike protein, a few minor modifications, and you can put that into various different vaccine development pipelines. But I think what we kind of maybe didn't quite appreciate at the time is, obviously, we're designing a vaccine against the very, very first example of the virus, which is a virus that's only been in human populations for, a couple of months maximum. It's been in some other animal, most likely bats, possibly via another animal, an intermediate host. But effectively, the virus is just at the very start of its evolutionary journey in terms of adapting to humans. And what we've seen over the period of about a year is some very clear evidence of convergent evolution. So the same mutations in the spike emerging multiple times in different places, but presumably to have the same kind of impact. And we think that some of those mutations do change how well that initial vaccine works. So I think, though, that so that's in a way that's a pity that we've designed against that first version, but it makes sense. We had to get going. What we do have a great opportunity now is we have a really good database of the important spike mutations and the common spike mutations that are circulating. It seems very likely that it should be quite practical and easy and fast to update the vaccines to incorporate those mutations that are widespread now internationally, particularly one called E484K. That's the one that we really would like to target. And of course, we also have the mRNA vaccines, which are effectively new, and in theory, replacing or adding extra gene sequences for spike into that vaccine should be a very, very straightforward thing. So I think we don't need to necessarily have a completely different type of vaccine, but we almost certainly are going to have to update it to represent that more humanized, if you like, human adapted spike gene sequence that we're seeing internationally.
Thank you very much, Nick. And I was going to ask you actually about the mRNA vaccines, because they are designed to basically create a spike protein, and presumably it's impossible to have an mRNA vaccine which would create an entire virus, or at least the entire outside of a virus.
Well, you wouldn't want one that created the entire virus.
No.
but yeah, I suppose there's no reason you couldn't incorporate other protein domains that, if you thought they were particularly antigenic, into an mRNA vaccine. I don't know if anyone's working on that.
Thanks very much, Nick. Sorry, Alan, you were going to say something as well.
No, just two little points I wanted to add is one, the spike protein has proven to be an exceptional target for the vaccine. You can see that from the early data showing how successful the vaccine is. Nick alluded to Israel. The other thing that's been a benefit of just targeting spike is something that vaccinologists really tend to try and do is be able to differentiate infected from vaccinated individuals. And so by just targeting spike, that means that you can do serology tests, for example, on other parts of the virus, like nucleocapsids. So you can differentiate infected people from vaccinated people. And it's a massive benefit to be able to do that. Certainly in veterinary virology, any veterinary viral vaccine always has to be able to differentiate infected from vaccinated. So there are benefits to just choosing one target.
That's really interesting. Thank you very much. Right, I'm going to move on to the next question now, which is coming from Rebecca Westaway, who I believe is with us and able to ask her question, not answer it. We're going to ask the panel to answer it. Rebecca, are you there with your question?
Yes, I am. Can you hear me?
Yes, you can. Go ahead.
Lovely. Is it ever likely that there might be an undetectable variant? And if so, what solutions are being developed to overcome such a situation? Thank you.
Thank you very much. What do you mean by an undetectable variant? Could I just ask you that?
That would not be detected by the methods that we currently have. So whether or not that's a PCR or lateral flow.
Okay, over to you, I think, Alan. Could there be an undetectable variant?
It's a great question, and obviously Nick was heavily involved in this. One of the major reasons that the so-called UK variant B117 was detected was because a gene that's used in the PCR test actually failed as a result of one of the mutations. But I think, coming back to answer, the beauty of most of the PCR approaches that are used, certainly in the UK, is that you don't just look for one gene. So the PCR test that Alex and I use in our testing lab, we actually look for 3 genes. So as long as you have a really robustly designed testing strategy, the chances of enough mutations happening in all of the targets fall out of the test is really quite low. And then also, if you use the genome surveillance that Nick and the COG guys are doing constantly in the background, looking for new variants and new mutations, we should be able to stay sufficiently ahead of the virus such that we never completely lose sight of it. It's a good question though. I mean, it has happened in the past. There's a sexually transmitted infection, chlamydia, that years ago was PCR testing was done for that infection. And years ago, the one gene that was used to target in that test, a mutation occurred, and the test stopped working. And there were hundreds of thousands of chlamydia cases went undetected because the PCR stopped working. I think we've learned from that lesson. And as I say, the fact that certainly in most UK testing, we target 3 parts of the virus means that it should really be quite unlikely that it happened.
Thank you, Alan. Nick, can you add to that?
No, I think that's absolutely right, what Alan said. I mean, sometimes you hear that people say, oh, you know, the virus might be evolving to kind of hide from the diagnostic test, which, you know, is, but the reason that the diagnostic tests, for example, in the case that Alan talked about, where it was the spike gene actually we couldn't be detected by PCR. The reason for that is because a small part of the genome was deleted. So a few nucleotides that make up the genome were removed. That's the kind of reason that a PCR could fail. It would be incredibly good if the virus ever got to the point of trying to evolve to escape the diagnostics, because it would mean that our diagnostics were so effective that there was some selection pressure on the virus to actually escape from our diagnostic procedure. Sadly, we're nowhere near that point where that would be any kind of selection pressure. So, but no, I agree with Alan. I don't think that's a worry. I do think there's a worry, though, more generally about going to get very fixated on SARS-CoV-2, you know, and this is a classic error. You know, we're going to think about this for a long time now, and every time we see patients with respiratory infections, we're going to think of SARS-CoV-2. We have to always keep in our mind that the other viruses are available and will emerge, and our diagnostic testing is very focused on targeting things that we think are likely, and that can change over time. And we have to be always mindful that other viruses, new viruses, or altered versions of viruses we know about can be circulating, can be emerging all the time. So, I mean, one thing that we look at in Birmingham for that is a process called metagenomic sequencing. It's a research interest of mine and also Alan's, where you don't have any presupposition about what you're looking for. This is how SARS-CoV-2 was discovered. You just take some body fluids and you sequence all of the RNA and DNA in that sample, and you see if you can find anything that looks weird. That's how they found SARS-CoV-2. We don't do that routinely in the UK, but we should do. We should be on the lookout for new things all the time, because we know this has been a great lesson about how new pathogens can emerge very, very quickly. The quicker we detect them, the more likely we are to be able to suppress them at source and not end up with an epidemic or a pandemic.
Thank you very much, Nick. Moving on to the next question now, which is coming from Holly Quinton, who's a GP, and she says, I'm struggling to convince some of my patients to have the vaccine, as they say, with new variants springing up, they're worried about the efficacy of it. Are there plans for genomic alteration of vaccines as we approach autumn next year, which harks back to one of the earlier questions? Also, at the moment, asking GPs to take bloods for antibody levels, it's perhaps not logistically possible. So I wonder if I could direct that to you first, Alex.
Oh, Holly, I completely agree with you. And that's why antibody testing's got to be done within clinical studies that are properly set up. And, you know, if it is reassuring, there is a whole gap analysis nationally looking at all the vaccine studies to make sure that we are doing antibody testing in, you know, the right age groups, the right comorbidities, the right ethnodemographics, so that we can get a really good look across the country of what antibody responses are. I very much doubt that antibody testing will routinely come into primary care, except there's going to be big pressure for it. I don't know what you're finding, but I'm getting patients emailing me or ringing my secretary every day saying, I want to know what my antibody level is. I want to know whether I've responded or not. And it is a pressure, and I know that pressure is going to be there, you know, in primary care. So I don't think you'll need to be doing that testing regularly, but it may be odd cases where you may want to do it. And I mean, I'm just thinking about some of our immune deficiency patients in the hospital. Whether they've responded or not to vaccination may be relevant. I was having, similar to a discussion I had with a rheumatologist this week about the chickenpox vaccine, and they've got an immune deficiency patient who doesn't have antibodies against the varicella zoster virus, which causes chickenpox. And so there may be some cases where you want to check somebody's antibody status, say before they went into chemotherapy or before they went into transplant. But I think for routine antibody testing, that's unlikely to fall into primary care.
Thank you very much. Nick or Alan, do you want to comment on that one at all? We'll move on to the next question then, which is a question from a student regarding lateral flow testing on campus, Joshua Kearney. Joshua, are you there and able to ask your question?
Yes, I am. Thank you everyone for your time today. I'm just interested to hear about what we've learned or what we're learning from the lateral flow testing that's taking place on the Birmingham campus, and what implications this might have beyond just campus safety.
Thank you very much, Alan. This might be a question for you.
It's a great question. I think that I've actually found being involved in the lateral flow testing really interesting, and we are finding out some really key things. There's a lot of debate about lateral flow tests and how good they are, how bad they are, when they should be used, when they shouldn't be used. I think the data that we're generating, so we passed 10,000 lateral flow tests last week this year on campus, and we've been collecting lots of data in the background around those lateral flow tests. I think the thing we can safely say is that certainly in our hands, we aren't seeing false positives. There's been a lot of talk about lateral flows produce false positives. We've taken any lateral flow positive we find, we've taken to the lab that Alex and I run, and we've done confirmatory PCR, and they're positive, and then we pass them to Nick, who genome sequences the viruses just to check that we don't have anything nasty on campus. So in our hands, the lateral flows work really well. I think the other side of it is that of those 10,000 tests, we've detected 50 people who were completely asymptomatic and who had really high levels of SARS-CoV-2 infection virus. And we've managed to find them and notify them they were infected and isolate them and break. That's 50 transmission chains we've broken on campus. Now, regardless of what you think about lateral flows and how they should or shouldn't be used, it's very difficult to argue with how, basically how successful that is as a strategy. If we weren't doing lateral flow testing, that would have been 50 transmission chains running through campus, and we'd have had to, impose measures like we had to in September and October. So I do think they're massively valuable. I think the other thing we've found since we started the testing again in January is that we ask people if it's the first test or if it's a follow-on test, and the data is really clear. The vast majority of our positives are people who attend for a first test. So people who are coming back for regular testing, far less likely to return a positive result. And so to my mind, that suggests that continuous testing by lateral flow works. It reduces the risk of SARS-CoV-2 transmission in the setting where you're doing the testing. And this is all data that we're going to try and write up soon, just to add to data to the debate. Sometimes the debate gets a little bit heated and maybe not driven by data. So we're going to try and get that data out very soon. But I'm a huge advocate of it. There are things I wouldn't use it for. I remember there was a suggestion last month that we could use lateral flows to allow people to enter pubs. That's ridiculous. Lateral flow testing should be used as a red light test, not a green light test. But by that, what I mean is you use a lateral flow to support an activity that you have to do. You shouldn't use a lateral flow test to do an activity that would be lovely to do. And I think as long as you get the use case right, which is something Alex and I always rant about, as long as you get the use case right, lateral flows are a fantastic tool.
Some really important points there, Alan, especially about the change of behaviour if you get a negative on a lateral flow test. And in fact, there was a question that came in from Simon Hodes, I see in the live Q&A. He said there's obviously many concerns about false negative rates with lateral flow tests. Are we giving false reassurance by using them widely as a screening tool in terms of people behaving as if they're negative? surely the LFTs have their place, but maybe not for mass screening. So how would you respond to that?
I think it's a great question, and I agree that I don't think they should be used as a mass screening test in terms of everyone takes one test and then you go off on your merry way. I think they don't detect very early stages or very late stages of infection, and obviously the worry there is where it's not detecting very early stages. That's why, you know, the numbers I suggested about people who are coming back for follow-on tests, if you're repeatedly testing at relatively short intervals, then the fact that you're not catching very early stages of infection isn't so important. There's quite a lot of published data now showing that at that very early stage of infection, you're much less likely to be infectious. So there is some correlation that where lateral flows detect viral infection is around about the stage where you're most likely to be infectious. So I think that continuous testing with lateral flows is what's really important. They should never be used as a one-off, I'm good to go. That's something I'm very adamant on.
And do you think that they are adequate in the hands of parents? Because obviously with school testing now, that's been handed over to parents to perform the tests on their children.
It's a really good question. We currently have some at home to test us because I have a little lad in primary school. We don't have to test him, but we're supposed to test ourselves just to check he isn't bringing it home. I think the beauty of the lateral flows, I think what we've learned is, you know, I get a lot of criticism that the reason our figures look so good in campus is because we've got myself and loads of PhD students doing the testing, but actually, I don't think that's the case. The lateral flow testing is very easy. It's very simple. And I'm not sure if there is any data to show that, you know, parents administering it rather than a trained healthcare professional makes any difference. And I think actually there's been some data recently showing that, you know, when lateral flows first came out, it was you had to go as far as you could up your nose until basically your eyes were watering, and then you had to make yourself vomit going as far down your throat as possible. Those guidelines have actually changed now because it does seem like, you know, the sampling does seem very efficient at picking up virus as it is there if it's there. So I'm not terribly convinced it makes. And again, regular longitudinal testing kind of negates any impact there might be on how good or how badly you swab yourself on any given day.
Thank you very much, Alan. Anybody else on the panel want to comment on use of LFTs?
Well, Alan and I have thought a lot about the use case, and this is something we've been discussing nationally with government as well. You know, we've got a whole suite of new technologies that have been developed, and actually we've been part of evaluating technologies here at the University of Birmingham. And we started by setting up studies looking at NHS asymptomatic testing as the cohort for the general population. And what we were trying to do is work out what is the best test. And actually, Alice, the best test isn't just the test that has the best performance characteristics. Because if you have a test that is unacceptable to the person doing it, i.e., you know, they don't want to do it every day because it hurts sticking something up your nose every day, or, you know, they just don't find it acceptable, they won't do it. And so you won't get results from it. So it is not the best test. If you've got a test that is easy to sample, but is really intensive in the laboratory, one of the national shortages we have in the UK at the moment is biomedical scientists. We just do not have enough of them. We've under-resourced pathology in the NHS for years, and suddenly we have none to be doing all this testing. So if you need lots of really highly skilled laboratory scientists doing the test, again, it's completely unsuitable for large population testing. So actually the best test now is one that is suited to that situation. And I think the other thing that we learned really early in the pandemic is, a bit political here, but by centralizing and only going to one source, actually if something goes wrong with that supply chain, we're scuppered. And actually, so diversity in testing and mixed models of testing. So, the government reached out to higher education, and that's where Alan and I set up the turnkey lab here. So, you know, the NHS couldn't do it all by itself. We needed, you know, private institutions. We set up the Lighthouse labs, we had higher education. And so a combination of testing and different types of testing to suit the situation. And for mass testing, or for large cohort testing, I should say, like at the university, actually the only way we could have done this, the only way we had the logistics to do this was with lateral flow. And, you know, we know it's not a diagnostic test. For the diagnostic test, you want the most sensitive test, which is, you know, the PCR. But in terms of case detection and in terms of reducing transmission, you are picking up the individuals that have the highest viral load. And so it does do that purpose.
And Alex, I wonder if I could just push you a little bit further on that, because at the moment it's voluntary for parents to perform those tests on their children at school. Would you advise that they should do that? This is helpful to the whole programme?
That's a question of ethics, Alice. And you know what is our, I don't know, civic duty? I think I've been discussing with my teenage daughters recently, and you know, I would encourage people to test. The reason to do longitudinal testing is that you improve the sensitivity of your test. So this is something brand new and a brand new concept to me as a, you know, diagnostician, if you can call it that, is that actually the sensitivity of your test depends on how often you test and whether you are testing at the right time. And what we found with repeat testing, and if you know, we're looking at the testing interval, if you test once every 2 weeks, you have a certain test sensitivity. If you test every week, you get a certain sensitivity. And I think this links back to what Alan was saying. Remember, with a virus, it's got to replicate, and so it exponentially rises, and then you get unwell, and then it goes down again. And that's over a few days. And if you can pick that interval where someone is producing virus, but is not yet unwell in themselves because their immune response hasn't been generated, that is your window for testing. And what we've found through all the asymptomatic pilots is that is about twice a week. And so if we are doing this twice a week, we're getting a much more sensitive test.
Thank you very much, Alex, and some really interesting comments coming through in the chat as well. Rachel Adams said with the guidelines from their school for LFT was that eye watering and retching were expected. So it's quite nice to know that you don't have to make somebody retch in order to get a decent test. But she also says, which is lovely, her 12-year-old and her friends are all completely chilled out about it and finding it really easy and actually better at doing it than their mother is. So that's fantastic. Right, another question coming in now from Brian Kirkland, who I think is there and ready to ask this question. It's a question about patterns of infection through time. Brian, are you there?
Yes, I am indeed.
Go ahead.
Okay, well, my question is about variants. I assume that by now there'll be quite a number of variants of the virus in different populations around the world. But a lot of them we won't be aware of because either the countries don't do any testing or they only do a limited amount of testing. If a new variant evolves which is more deadly than the existing forms or much more contagious, somewhere in a population in Africa or Asia or South America, away from the big population centres, how quickly can we expect to pick it up and react to it?
Thank you very much. Great question. As we know, we are doing quite a lot of work on that in this country. Nick, would you like to start with that one?
Yeah, I think it's a really interesting question. It's something that we're quite preoccupied with because you're absolutely right. You know, our ability to detect variants is really going to depend on how good our genome sequencing capabilities are. And of course, in the UK, we have really very good genomic coverage. We're getting to the point where we're looking at sequencing almost half of every case. That's helped by the fact that the cases, half of all the cases, which is helped by the fact that the cases have come right down. But most countries are not like that at all. And in some countries, quite notably, you'd expect to be really good at genomic surveillance and not very good. And the US is a good example of that. It really took a long time for them to get up to speed because of reasons. What we're finding is that, certain variants, that we are detecting quite a few new variants through sequencing of imported cases. So a good example of that would be the P1 variant, which is sometimes called the Brazil variant. That most likely emerged in the Amazonas region in Brazil, but it was detected through travelers from Brazil arriving in Japan. Japan is a really well-connected travel hub, but they also have a policy of testing all inbound travelers and doing genome sequencing. So they detected the Brazil variant in Japan. And later on, we did some work in Brazil, helping Brazilian researchers with their sequencing to detect it locally. And the UK now has a program of managed quarantine facilities, whereby any person coming to the UK from a red list country, of which there are quite a few now, will get a day 2 and a day 8 test, and if either of those are positive, the genome will be sequenced as well. So that's giving us quite a good view of countries that are connected through international travel, which is obviously, most of the world actually. So, but it's not as good as having direct local genomic surveillance in each country with good coverage. So we are a little bit lagged on detecting these variants. It might take a month or 2 to detect a variant before it's where we have sufficient cases to be able to detect it through international travel. Lots of groups, including in Birmingham, are working on packages of protocols and methods and tutorials and training courses to try and increase the coverage of genomic sequencing worldwide. So we work with groups particularly in Africa and in South America, but it's a definite focus of the funding agencies to try and ensure that all countries are represented by genomic surveillance. That's a project for the coming year.
Nick, that's really interesting. You just answered the question that I was going to ask you, which was that whether with all the expertise that we have here in the UK, and especially in Birmingham, actually, whether we are helping other countries to grow their capacity, and that's fantastic to hear that is indeed happening.
Yeah, one of the best countries actually in Africa at sequencing was the DRC, and partly that became out of the fact that we and other groups have worked with DRC to build their genomic sequencing capacity to help with their Ebola outbreaks. So having that capacity there for other infectious diseases means it can be reused easily for new pathogens. So it's really important to have that platform.
Fantastic. Alan, did you want to add something there?
Yeah, just really briefly. I think, Nick's pioneered this, but I think it's really important that we get away from this sort of Western-centric idea that it's only a handful of big Western countries that can do genome sequencing and it should all come to us. The model has to be to widen it out. Every country should be able to do this. And it's kind of what makes even more galling is that lots of members of the public will have missed this, but last week the government slashed funding for overseas research. So there's loads of research projects like Nick describes, where we use the money to train people in low and middle income countries in how to do things like this. And that budget was essentially cut overnight last week. And that kind of research is absolutely vital. Research shouldn't be about doing cutting edge, expensive research in big, shiny new buildings. Research has to be on the ground in countries where it makes a difference. And I really hope that we can get a rethink on that strategy. Otherwise, there is going to be this over-reliance on a handful of countries doing it all, and it doesn't work. It has to be local. It has to be regional.
Thank you very much, Alan.
That's so true for vaccine studies. And there's a lot of vaccine hesitancy in some ethnic groups has been really how we've used a lot of lower income countries to run vaccine studies in the past. And it's that you've got to have that local engagement with research to be able to deliver these kind of projects. Otherwise, there's going to be national suspicion. And certainly, the MRC and Wellcome have been doing these projects, around the world. And it's how we get that local engagement. It's not just the big shiny machines. It's the people on the ground actually doing the research. And if we don't support and fund that, actually, we're going to dry out of opportunities to be doing this kind of big vaccine research nationally, globally.
Yes, thank you for that, Alex, and also that that point that we have disinvested in public health as well, particularly in this country. So we seem to be pushing all of our funding towards the high tech end of things and forgetting about public health, which is unfortunate. And we had a couple of comments coming in from the Q&A as well on these points. People saying, really great point. Thanks for highlighting. I mean, that question of cutting the funds, Alan, that sounds like a really retrograde move. Is there anything that people can do to try to change that or to bring that funding back?
I think make as much noise about it as we can. The universities, like all universities in the UK, are trying to lobby government to try and reverse the decision. I don't think we'll reverse the decision. I think what we have to do now is make sure that there aren't further decisions taken that hit that type of research even harder. But it's happening now. There are projects that were about to start that are now being told they won't be starting. And it really is a backward decision. SARS-CoV-2 didn't start in Kent. It started in the middle of rural China, and there's lots of other potential pandemics that we've dodged a bullet on, which started in low and middle income countries, in animal, human, close interaction reservoirs. This is where the problems lie. It's not in big shiny hospitals. It's in rural, low and middle income countries, and we have to make sure that we're doing the research in the right places and training the right people.
Thank you very much. Moving on to the next question now, and I think there's a question coming in live from Valerie Spear. Valerie, are you there? Oh, I thought Valerie might be there and ready to join us. Mike Fletcher, have we had Mike's question? Mike, are you there?
Sorry, I hadn't unmuted myself.
Oh, Valerie, you're with us. Fantastic. Ask your question.
Sorry, I hadn't unmuted myself. My problem. Are we any further forward in understanding why there's such a wide spectrum of response to the virus in the human population, and in particular, why children don't get symptoms? And this might also relate to, could this change with the emergence of new variants? My other thought was, are there any other viruses that have this kind of profile? But I'm not aware of any that don't affect children but affect the rest of the adult population.
Really interesting question. Thank you for that. Nick, I wonder if I could come to you first for a response.
Yeah, I don't understand exactly why children, so clearly have much less of a severe response to adults, and particularly older adults. There is some really interesting research out in the past few days, thinking about why the new variants seem to be more transmissible, and the UK variant also, there is now quite good evidence that it's more pathogenic, i.e. can cause higher mortality and more severe disease. And the paper in question is thinking about the innate immune system. So it's thinking about a class of molecules called interferons, and that's our body's, I'm not an immunologist, but that's our body's way of being able to detect invading viruses, invading bacteria without any prior knowledge of them. So it's a system that can recognize certain features that are common to pathogens, but not to components in the human body. And so, recognizing things like the viral genome itself and certain structures associated with viruses. And I think that's a really good theory, talking about why the new variants have this difference. We know that viruses will there's always a pressure on a virus to be more transmissible. Anything, by definition, any virus that can transmit more easily to more people has an advantage over a virus that doesn't, because there'll just be more of that virus around. We know that the Kent variant is more transmissible, and I think that some of the mutations associated with the variant could well be associated with this innate immune response. And if we look at the other side of it, we look at host genetics. So host genetics are important, and in anyone's response to any given pathogen, your particular genome will have some kind of impact. And when we look at associations between severity of illness and host genetics, again, these interferon family of genes come up as our kind of most striking hits. So I think part of the answer to your questions is something to do with innate immunity, is something to do with recognition of the virus, and it may be that the new variants are better at evading that innate immunity, our ability just to recognize that this is some kind of virus, and the body should mount a kind of broad immune response. So quite different from the idea of adaptive immunity and T and B cells and all that stuff. But I think that might be part of the answer. And Alex will know much better than me about the difference between the innate immune response in children and adults. But maybe that's part of it as well.
Thank you very much. Yes, Alex, would you like to comment on that?
It's a really interesting question. So over the last, year, I mean, the key things that I've learned are that the older you are, the more likely you are to get sick. That obviously some of the ethnic populations are more at risk. And certain comorbidities make you more at risk. So hypertension, maybe diabetes. But then the other big thing that's come out is obesity and weight. And that, most of the patients on ITU that I've seen have been, pretty overweight. So that brings into question, not just because when I think about risk of an infection, I think about the host, and for the host, it's their genetics. I think about the environment and what they're being exposed to and external influences. And then you think about the pathogen. So Nick's just eloquently spoken about the different variants. If we think about the environment and the host, there are different factors within the host, like differences in this innate immune response, and interferons are the main chemical signal that we use to protect ourselves against viruses. But also this ACE2 receptor that is important for viral entry. And we know that the ACE2 receptor is slightly different in different populations. And the age thing's interesting as well, isn't it? And I wonder where that links with some of the other environmental factors. Like children are less likely to be overweight than adults. So that might be a factor. The cytokine signature in the studies that we do have on children versus adults show slightly different, chemical signals going on in response to infection. And I guess the other thing is ethnicities. I was looking the other day at, the mortality rates and the severe illness rates in the UK, in the Bangladeshi, the South Asian, the Indian and the Pakistani populations. And then I was looking at what that mortality rate was in Bangladesh, in India, and in Pakistan. And there's a real difference. And so that tells me that, okay, genetics might be important, but actually your environmental exposure may be more important. So I don't know why children haven't been so affected. It may be something to do with their density of ACE2 receptors. It may be something to do with their interferon response. It may be to do with something that they run around and they keep the weight off. You know, I've seen interesting studies looking at, I'm sure all of us have heard about cholesterol reducing drugs, statins, but there's a different class of drugs, fibrates. And there is some evidence that fibrates stop the viral transmission into cells. And you wonder whether that's to do with how it disrupts and looks at the fat molecules in the body. So there's a whole group of researchers looking at fat, really. And, you know, fat just isn't an inert subject. It's a highly immunogenic substance and part of our body. And so as our weight changes, actually that affects our immune system and our ability to fight infections.
Thank you very much, Alex. Well, we have got about 10 minutes left, and I'm going to try to squeeze in 4 questions. So questioners, I'd like you to keep your questions nice and concise, and panel, I know you'll be very concise and precise with your answers. So first up then, Mike Fletcher. Mike, are you there?
Yes, good afternoon, I'm here. I just want to preface my remarks by saying that it's amazing that 54 years after I did a BSc special in genetics, I finally found a use for the information. My question is, looking to the future, are there broader scientific spin-offs from the work that's been done to understand the pandemic, which will apply to other disciplines and other areas of study?
Fantastic question. Alan, would you like to take that one?
Very quickly, I'd like to think that the way that we've done genome level surveillance of infectious disease continues. Nick mentioned that I think everyone's become obsessed with genome surveillance, but for very good reason. It's phenomenally powerful, and we have to maintain that now. Nick and I have been doing genomics for a long time. We've been trying to tell the world how powerful it is, but it's always been seen as a bit of a niche topic. I'd like to think now that we realise that genome surveillance is the way to go for infectious diseases and lots of other things as well.
Thank you very much. Anybody else got anything to add? Yes, go on then, Alex.
Winter pressures, Alice. Every single winter, the NHS is overwhelmed by respiratory infections. And what we've learned about transmission of viruses in the community, in hospitals, about testing for viruses, about point of care diagnostics for viruses. I hope the legacy from COVID is that we no longer have to treat patients in corridors in the NHS.
Thank you, Alex. Moving on to the next question then, and it is from Graham Long. Graham, are you there with your question?
Yes. If I can
Ask away.
I've lost it, my apologies.
Can you remember it?
Yes. After the government relaxed the restrictions at Christmas, there was a huge spike in infections. Now that we're leveling out at 5,000 a day, and the lockdown relaxations are due to start from next week, would you recommend that the government extend the current lockdown until after the Easter school holidays?
What do you think, panel? Should the lockdown be extended until after the Easter school holidays? It's a tricky one. Gosh. It's quite political, isn't it?
Gosh.
We're starting to open up again a bit on Monday, according to the roadmap. But obviously the government will be looking at the facts and figures at the moment. We're seeing an increase in cases amongst school-age children, obviously.
I think this is such a difficult question. I do think that the government's roadmap as laid out is very, very likely to slip, I have to say, in terms of, I think some of the things that we are not allowed to do now, we possibly could be allowed to do. I feel like we could probably go outside and meet people, rule of 6, and that would probably be okay now the weather's improved a lot. And there's good evidence that outdoor socializing in small groups is not a very high risk activity. But if you look at the roadmap, it takes us quite quickly, I think in June, to basically a full opening and the nightclubs being open. And I know people are booking into music festivals. And I feel like I feel like that's optimistic. I have to say, I feel like that's an optimistic roadmap. The government have caveated, of course, by saying that that's the earliest it can possibly happen. Yeah, I don't know about whether we should extend the lockdown up to after Easter holidays, particularly. But I do think that I'm expecting that roadmap is got a good likelihood of slipping, quite honestly.
Thank you very much, Nick. That was extremely reassuring to hear Boris Johnson say that he was going to be looking at data, not dates. I mean, obviously there are dates there in the roadmap, but they are aspirational, and hopefully the government will be guided by the data. Right, I'm going to fit in another couple of questions. We're up against it, but Paul Cavendish, are you there with your question? Paul Cavendish. Oh, he's got a very slim chance of getting in on the call.
Hello, can you hear me?
We can.
Oh,
we
hi
can.
there.
What's your question?
Good afternoon. I was having a COVID conversation with a friend the other day. She's a healthcare worker and she's had the COVID infection twice. But she's had a more recent blood test and they found no circulating antibodies. And we were wondering, well, is it worth having the vaccine on that basis?
Alex, is that one
Paul,
you can
I'm happy
answer?
to take that one. So there's some things we'd need to know. We'd need to know which antibody test that she had, because a bit like we've talked about the lateral flows and the PCRs, some of the early antibody tests were of really low sensitivity. And we have antibody tests against different targets of the virus. So we'd need to know which type of antibody test that she had. And what I do know from our early vaccine studies, and I said at the beginning of the webinar, is the vaccine is so much more immunogenic than natural infection. So I would absolutely encourage her to go and get her vaccine done. But Paul, we know that some people are going to not be able to respond to vaccination. And, you know, the patients that I look after in my day job are patients with immune deficiency, with antibody deficiency, and they don't produce antibodies. And so we know that there is going to be a subgroup of people. So it's very difficult to give, you know, personal advice over a webinar, but I would absolutely go for the vaccine. It is somebody's best chance of protection. But if there is a problem with recurrent infections, it's always about, is there do they need a checkup basically at the GPs and a look at their immune system.
Thank you very much, Alex. I'll just squeeze this one in. Why did all the money spent on test and trace not make a difference? Who would like to take that question? It was a lot of money.
Yeah, can I jump in on that one?
Yeah,
and
do.
For the next 40 minutes. My own personal opinion is that we set up a new testing system, which we had to do. We weren't testing enough people this time last year, and we had to increase the number of people we tested. A decision was taken that we then had to set up a new testing infrastructure. And unfortunately, from the start, that wasn't connected enough with existing public health infrastructure. It wasn't connected with existing public health teams who are brilliant at doing infectious disease tracking and surveillance and epidemiology. And that's why it didn't work for the money that was put in. I still maintain the testing part of Test and Trace was a huge success in terms of what it delivered, the quality of the tests it delivered, and how many tests it delivered. The problem was that it was never connected to something that could then action the test results. And I think that's where the big problem was with it.
Thank you. One last live question then, and it comes from Gavin Pearson. Gavin Pearson, you have the floor.
Hi there, can you hear me?
We can, go
Hi.
ahead.
Yeah, thanks. I was just wondering, is all your research and your work done in isolation, or do you work with other universities and institutions?
That is a great question. Are you just there in the ivory tower of the University of Birmingham, sealed in, not talking to anybody else around the country or indeed the world? Nick, are you completely isolated?
Well, I think this has been a really great example of scientific teamwork, actually. One of the more positive things from the pandemic has been putting aside some scientific rivalries to throw in to a national and in an international effort. For our part, the genome sequencing work is a consortium made up of about 17 universities, the 4 public health agencies in the UK, the Sanger Institute, and the NHS, and we've been working hand in hand, and we're effectively have formed this kind of huge virtual research group, you know, hundreds and hundreds of people coming together to both generate those data sets and to analyze those data sets in lots of asking lots of different questions of that same data, and that's really paid off in terms of our understanding of the virus evolution and our ability to you know, quickly detect these new variants of concern. So no, I mean, it's not perfect. We're much better at working on a national level, I think, than an international level. We still have this challenge about how to integrate genomes from across the world and make sure that each of the different countries can benefit from the information in there. So that's the next big challenge for us. But certainly from a UK perspective, it's been a really strong collaboration, and I think, you know, we've made new collaborations that will persist and new structures that will persist going forward for other infectious diseases. So that's been great.
And very briefly, I presume that Alan and Alex, you agree with that. It's a huge collaborative effort. Thank you very much. I'm going to have to draw it to a close. Thank you for the absolutely great questions that have come in today. And thank you to our amazing panel, Professor Alex Richter, Professor Alan McNally, and Professor Nick Loman. I'm sure everybody who's watching appreciates the time that you've given us today to share your expertise. And thank you everyone who is watching this Bringing Birmingham to You event on mapping the virus. And as well as events like this, Alex, Nick and Alan, I know, are all on Twitter as well. So you can follow their work and engage in conversation there as well. We will continue with this series of webinars online, and we'd love to hear from you about any areas of research you'd be particularly interested in. I know some of you have already shared your ideas with us today. Please, if you haven't, then do share your suggestions with my colleagues in the alumni office, and let's thank them too for organising this event today. So that's it from us for now. Thank you again, everybody. Stay safe, keep well. Goodbye for now.